So we'll be talking about the small intestine. The small intestine is the most important part of the digestive system. Um, Everything else is less critical. You can live without a stomach, you can live without an esophagus, you can live without a large intestine, but the small intestine is where the majority of things that you need to survive happen. So it's the major organ of digestion. Most of the digestion happens, and most of the absorption into the body happens. Remember that the lumen is outside the body, so getting stuff into the body happens there. It's a couple of meters long, up to 4 meters long. It starts at the pyloric sphincter and it ends, the terminal part is the ileocecal valve. It's subdivided into three areas: the duodenum, which is right at the from the pyloric sphincter, and it's about 12 inches long and is found retroperitoneally. The jejunum, which is the next major portion, it's attached to the body walls by a mesentery, and it jejunum means twisted. Ileum means empty, and so that's the terminal portion. The last portion is the ileum. It starts here. The duodenum comes around in a C shape, convex to the right. The pancreas sits right there in the duodenum. Then this is the jejunum, all of this, and then the ileum is this part, and the ileum ends at the ileocecal valve at the cecum of the large intestine. So in the duodenum, its job is basically to mix things together. So this is where the bile duct and from the liver, gallbladder, and the pancreatic duct from the pancreas squirt their contents to mix with the chyme coming from the stomach. So there is a spot in the wall of the duodenum called the duodenal papilla, where these joined bile ducts and pancreatic duct squirt their contents out into the duodenum. We call the area where the two ducts join the hepatopancreatic ampulla, and there's a sphincter there that controls what leaves. It's called the sphincter of Oddi, or the hepatopancreatic sphincter. Both names are the same. So it works like this. Along here, all these exocrine cells of the pancreas are making the digestive enzymes. It's very, very alkaline. It's as alkaline as the stomach is acidic, and it passes along this duct, the pancreatic duct. The main pancreatic duct continues on and joins here with an ampulla. There's a sphincter here. There's a second duct that is kind of a back door. It's in case this one gets blocked, because you don't want the pancreas to digest itself. And that's called the accessory pancreatic duct. The ducts that leave the liver, each one leaves each lobe of the liver, so the left and right hepatic ducts are here. They come together to form the common hepatic duct. The common hepatic duct at this point changes name to the bile duct. This duct that leaves where the common hepatic duct becomes the bile duct is called the cystic duct, and this is a two-way tube that goes to the gallbladder. So when this sphincter is closed, bile backs up here and goes in and it's collected up into the gallbladder. When this is open and the gallbladder's muscular wall is squeezed, the bile comes out and goes down this bile duct here and squirts out into the duodenum through the papilla here. This is a place where gallstones will get caught and cause cholecystitis. Very painful in here. So along the small intestine, there are modifications that are designed to increase the surface areas to increase the amount of absorption. That's the primary job, is absorption. So the walls of the intestine are thrown into folds called the plicae circularis, which means circular pleats or circular folds. And then the surface epithelium is thrown up into little fingers called villi. And it ends up looking like terry cloth, like towels, as it were. Now, each cell on the surface of the villi, each columnar epithelial cell, is got its apical surface thrown into folds called microvilli. All of these greatly enhance the surface area for absorption. So the circular folds, they're there. They cause the chyme to spiral and to move. They slow it down so that it doesn't pass too quickly. There's enough time to mix and with the digestive enzymes and start to be digested. And it looks like this. These are the plicae circularis. Each plicae circularis is got villi on it, and then the villi have microvilli on them. There's also all kinds of goblet cells in this epithelium because it creates a fair amount of mucus because you want to keep on lubricating the passage of this material. We call the microvilli on these cells, these absorptive cells, the brush border, and they actually have integral proteins, part of the cell membrane, that act as enzymes, and we call them the brush border enzymes. And it's important to note that these enzymes do not actually mix with the food, with the chyme. The chyme touches the brush border enzyme, the enzymatic reaction happens, and then absorption happens immediately there. The brush border enzymes are responsible for the final hydrolysis, the final chemical breakdown of the food molecules. Now, there are these ideas of intestinal crypts. What happens at the base of each villi, there is a kind of a deep pocket where there are secretory cells, there are enteroendocrine cells, there are lymphocytes in there. There are antimicrobial agents, and most importantly, there are stem cells. This is where the new epithelium comes from. This epithelium, especially up here, has to be replaced all the time. So we have the stem cells down and protected in these little holes, in these little crypts. Here, each villus has got a blood vessel, like a venule, and an arteriole, capillary beds for absorption, and there is a lymph capillary coming down to a lymphatic vessel here. The water-soluble nutrients get taken up into the blood when they're absorbed here. The fat-soluble ones end up in the lymphatics and are returned or brought to the bloodstream via the lymphatic vessels, ultimately up in the thorax. The submucosa is specialized here too, because there's a fair amount of lymphatic reticular tissue. Uh, and they are involved with protection, especially against bacteria, but against anything that could be coming in. They almost act kind of like lymph nodes of the um, intestines. However, they don't have a capsule or trabeculae, so it's just basically the reticular connective tissue. And these are called Peyer's patches. There are also glands that secrete an alkaline mucus uh, to to further get rid of any acidity that might be in the chyme, but also to kind of lubricate the movement of the chyme along the intestine. There's secretions called intestinal juice that it's mostly water, um, and it's all about creating an environment to facilitate absorption. It's really about um, dissolving these water soluble food molecules, providing enough liquid to dissolve it to facilitate the diffusion into the blood. It's isotonic with blood, so there is really no osmotic gradient here. It just it's used to facilitate that. It's a fallacy. A lot of people think that intestinal juice has the enzymes that are involved with digestion in it. It's not true. The enzymes come from the pancreas and the brush border enzymes, which are not secreted but are actually part of the cell membrane. The liver is this huge accessory organ. Uh, for the longest time, it was considered that there were three lobes to the liver: the left, right, and caudate. Now they've decided to subdivide the caudate into the caudate and the quadrate lobe. So we talk about four lobes to the liver. Um, it's semantics, and that's all. Uh, but what you'll see in a lot of old textbooks and a lot of things that you may note, if you're looking on the internet or anything, if you're doing anything with liver, you will see that it's mentioned that there are three lobes often. It's been fairly recently, in like the last few years, that it's officially been considered that the quadrate lobe is a separate lobe to itself, and so now we talk about four. Um, it's suspended uh, from the diaphragm. It's up on the right hand side. It fills the whole hypogastric or hypochondriac region of the right side, right from the back to the front, from the diaphragm down to below where the costal cartilages are. Um, there's a broad ligament that separates the left and right lobe and suspends the liver, and that's what the liver hangs on. There is a round ligament called the ligamentum teres that runs down to your belly button, and it's basically the leftover remnant of your umbilical vein, because in a fetus the umbilical cord goes into the belly button, the umbilicus, the umbilicus, and the blood that's returning to the baby ends up going right to the liver, like it's like the hepatic portal systems. When you no longer have an attachment to the placenta, then that atrophies and becomes a round ligament. It basically sits like this. And so it's a very large organ. I'm not going to expect you to be able to identify the parts. It's not really that important to me. The lesser omentum attaches it to the stomach, and we've already talked about the portal veins. We've talked about the bile ducts and the cystic ducts, etc. We've already looked at this picture. The liver is divided up into structures called lobules, and so these are the functional units of the liver, and this is where the hepatocytes are found. And basically, and we'll do kind of the Reader's Digest version of this, it's arranged with all of the tissue that does the functions of the liver around a central vein. And that central vein will drain into the inferior vena cava eventually. At the corners, at 3 of the 6 corners that each one of these lobules have, we're going to see that there is an artery, a vein from the hepatic portal system, and a bile duct. And so we call that the triad. So the bile duct receives bile, the portal arteriole comes from the hepatic artery, and the venule is from the hepatic portal vein. And there are capillaries in here that are very leaky. They're sinusoidal. And there's actually macrophages live in there called Kupffer cells. Without making too big a deal of this, basically blood enters the liver lobules here via the hepatic portal vein coming from the intestines, the digestive system. Here, the hepatic artery that is bringing blood from the oxygen-rich blood from the aorta, and this blood mixes and goes through these capillaries here, where it interacts with these hepatocytes, the cells of the liver. That blood is then transformed because of stuff going into these cells, coming out of these cells, and then it goes to this central vein, which ends up in the hepatic vein, which ends up in the eventually in the inferior vena cava and going into direct circulation. Now, between the layers of the hepatocytes here, there are, like on the basal surface of the hepatocyte, there is a little canals, little ducts that will eventually come into the bile duct. So anything that has to go into the blood goes off the apical surface into the blood. Anything that comes from the blood comes from the apical surface. Anything that's going to be excreted in the bile ends up coming out the backside, and there's a bile duct that will drain each lobule. It's a 3-dimensional structure, so there's a verticalness and as well, but all that really matters is this this central vein and these triads at the corners. So what happens in here? Actually, a lot of things happen in the liver. On my provincial or my national board exams, one of the questions in the physiology content was list and describe in detail 15 major functions of liver. It does a lot of things. It processes all the blood-borne nutrients, it stores the fat-soluble vitamins, it detoxifies anything that's in there, it produces bile. This is where albumin is produced that maintains the osmotic gradients of the blood, etc., etc. But as far as the digestive system goes, it's processing nutrients, storing vitamins, and producing bile. Now, bile is an important substance because it what it does is it mixes with the chyme and it causes emulsification of any fats that are in the chyme. Uh, the bile salts are involved in this. Um, and so what it does is it enhances the breakdown of lipids uh, and the digestion of lipids by emulsifying, by taking the globules of lipids and breaking it down into smaller globules, emulsifying. Now, also within the bile, there's the the leftover pigment from red blood cells. It's called bilirubin, and that's what actually makes the color of the bile. And it's just a waste product. It's gotten rid of. There's cholesterol, there's phospholipids, there's other electrolytes, all involved. Most of them get reabsorbed later. So so everything except for the bilirubin ends up getting reabsorbed and recycled. Now, the gallbladder is used um, to concentrate the bile. It stores up bile when we are not using bile, when we're not eating, and it absorbs water out of it so that it becomes a more concentrated form. And it squirts bile into the duodenum when we have eaten a meal, especially a meal that's rich in fats.